Okay, so it's that time. First thing I'm going to do is pass around the sign-in sheet. The next thing I'm going to do is pass back what I have for you, which is your exam for most people. And then for those that were not here the day before spring break, or the Thursday before spring break, I also have your a couple of your labs. If you were not here the day before spring break, you'll also want to pick up one of these, because this is the side chains that you will receive on the exam related to the amino acids. There was actually, in a sense, a 16-point curve on this exam. 9 points because I only had 41 points of answers. And then I curved it an additional 7. So basically you got 16 points. So Katie Atkinson, Macy Broadway. Yes. Katie Duran. Mackenzie Fresnita. Sam Holt. This one here. Addie Key. Zach Lunk. Grace Nelson. So basically curve your exam grade by 7 additional points over what you see. Andrea Palos. Corey Parker. Flor Perez. Lisa Sahib. Maya's not here. AJ Smithwick. Elizabeth Ford. Okay, so what we're going to be doing today, Janaya, I have nothing else attached, is we will be continuing our discussion of proteins. So your proteins start with the amino acids, and the amino acids are responsible for the protein. Now, because there are a lot of pictures, I have resorted to PowerPoint, and I will probably use a lot of PowerPoint from now on. But it is in large part due to the fact that there are a lot of pictures, and I think pictures will help in the understanding. Whereas before there weren't as many photographs that I could use, but now we're getting to where there's quite a bit that visually will help in the understanding. So the Thursday before spring break, I introduced the amino acids. Amino acids have a common backbone. They all have an amine group. They all have a carboxylic acid group. And then attached to the carboxylic acid group is an alpha carbon that has some sort of side chain. 19 of the 20 amino acids are going to be chiral, because they, on that alpha carbon, the carbon next to the carbonyl carbon, they have 4 different groups associated with it. So again, There's not 20 amino acids. 19 of those are going to be chiral because there are going to be 4 different groups attached to the alpha carbon, which is the carbon next to the carbonyl carbon. The only one that is not is glycine. Otherwise, they are all chiral. Now, again, the main thing for a carbon to be chiral is that it has to have 4 unique groups attached to the chiral carbon. Now, when we start talking about carbons that are chiral, in the case of amino acids, we can describe them as left-handed or right-handed. And basically, it is dependent upon how they move clockwise or counterclockwise. Now, how do we determine their handedness? What you can do is you can add up the molar masses of each substituent connected to the chiral carbon. So in this case, you have a carbon, which is 12 grams per mole, according to the periodic table. You have 2 oxygens at 16 grams per mole each. So if you add up the CO2, that gives you a total of 44 grams per mole in a mass. Hydrogen, according to the periodic table, is just 1 gram per mole. Your CH3 contains carbon, which is 12 grams per mole, and it has 3 hydrogens, each at 1. So the CH3 is 15 grams per mole. And then your nitrogen is 14 grams, according to the periodic table, each hydrogen 1. So that's 14, 15, 16, 17 grams per mole. So the first thing I did was just to simply add up the molar mass of each of the substituents attached to the chiral carbon. Now we're going to number them from heaviest to lightest. Oh, and this is the same compound over here. So I'm just going to copy this for a moment. Okay. Now we're going to look at them from heaviest to lightest. The heaviest is going to be the carboxylic acid functional group. The next heaviest is the amine. The next heaviest is the CH3. And finally, we're talking about the hydrogen. If you go from heaviest to lightest, if you draw it from heaviest to lightest, you will see that this one moves in the left-hand direction. And so this is L-alanine. In the other one, if you go from heaviest to lightest, you will notice that it goes in the right-hand direction. Therefore, this one is D-alanine. Now, nature has a preference. Biologically speaking, all of the amino acids that we work with are going to be L. Again, nature has a preference between L and D. And biologically speaking, we tend to work with L. Our bodies are made up of L. Now, it doesn't mean you can't have in your system any of the D. It just means that biologically we are not made up of D. But D could be for one of the, for example, one of the D is caffeine. Obviously, we can take in caffeine. So it is possible that you will have an amino acid that is D, but as far as your biological proteins, your biochemistry, it is going to be the form of L. Any questions about chirality and how you can determine whether something is L or D? You will notice that when we're trying to determine D or L with respect to our protein backbone, it's drawn a little bit different. The carboxylic acid is going to be north on the molecule, and the amine functions is to the left of the molecule. That's only when we're trying to determine which handedness the protein is that we put it in that format. If that amine group is on the right, it goes, it's going to have a different handedness. Questions? Biologically, all 19 chiral amino acids that are in your body that form proteins are going to be L.
So most are chiral, and you said the one that's not chiral
is glycine.
One question. But like, what does it mean for the molecule? Like, if it's left-handed versus right-handed, like, what's different about it?
What's different about it is how it, technically what is different is how it polarizes light. But it also does have implications from a biological standpoint. But as far as L and D, the main difference is whether it polarizes light. Now, L and D also, while I gave you an example of one of the amino acids that you can have in your system that is D, that's not biological, it's something you take in. Some of the D are hazardous to your health. It all depends upon what it is. Caffeine's not exactly great for your health, but so that's another aspect of them that is important to know. Did that answer your question? Okay. Here you go. Enantiomers have similar physical properties. The one property that I can guarantee is different is how they polarize light, but their biological activity is going to be different. And again, as far as nature, anything that is natural within your body, it's going to be the L amino acid. Okay, now proteins. So we've talked about amino acids. We talked about the amino acid. When amino acids combine, they form proteins. The process of combining two amino acids is the same as we've already talked about. In that, in this case, your nitrogen is going to attack at the carbonyl. Your carbonyl oxygen is going to temporarily lose its double bond, but then remake that double bond. And ultimately, you're going to kick out a hydroxide group. And also from the amine, you're going to lose a hydrogen. So what you would make with valine and cysteine is you're going to make what we have learned is an amide. Now, while that is an amide, this bond that was formed is called a peptide bond. This combination of valine and cysteine is referred to as a dipeptide because it has two proteins in the chain. I mean, it has two amino acids in the chain. So in other words, folks, you already know the chemistry because we did the chemistry in previous lectures. We made amide bonds in the previous chapter. The difference is now we're applying those amide bonds instead of to just organic molecules in general. We're applying that amide bond formation to the proteins. And when you apply it to amino acids, and when you apply it to amino acids, that means it's called a peptide bond. Now, one thing we talked about prior to break is that we don't often see these dipeptides in a neutral form. These dipeptides are typically going to be in a Zwitter ion form. So instead of seeing it written in this format, you're just as likely, if not more likely, to see that dipeptide written in the Zwitter ion form. This is still valine cysteine. Notice I don't name it as valine and cysteine. It's now valine dash cysteine. That's how you name a peptide, is by naming the amino acids that make up the peptide. Now, when we draw it, we always will put the nitrogen on the left. And the carboxylic acid on the right. This is because it is called a C terminal. End and an N terminal. End. So we talked about a dipeptide. Valine cysteine is an example of a dipeptide. It's made up of 2 amino acids. You can form a tripeptide, which consists of 3 different amino acids. And then we can form polypeptides, which the majority of things that we'll deal with are polypeptides. Now, folks, one of the things that we're going to find out is important is the order of your amino acids. If you change the order of an amino acid, you're going to change the behavior. And there is a formula that you can use to figure out how many potential variations there are. So let's say I have 3 amino acids. Then the number of variations is going to equal 3 prime, or in other words, it's going to be 3 times 2 times 1. So So in other words, we're dealing with 6 variations. If you had 4 amino acids, then your number of variations would equal 4 prime. Or in other words, 4 times 3 times 2 times 1, or in other words, we're looking at 24. When you get up to 8, we're talking 40,000 different ways that 8 amino acids can combine. And then for something larger, you can quickly see how the variations. That variation is important. You change any one of them in the tripeptide or the polypeptide, you're going to change its behavior. So what can all of these protein structures do? One type of protein is going to be, in most cases, considered to be a globular protein, which we'll talk more about. And it's going to be your enzymes. The enzymes are going to be there to catalyze all sorts of functions. An important one is amylase, and we'll explain, I'll get to how we've come up with the ending, ACE, a little bit later on in the semester. But anyhow, one is amylase, which is going to be the first step in digesting carbohydrates. Enzymes are used to start breaking this amide bond. That's one of the key things that they do with the proteins, is they will also break amide bonds. Another that we really won't talk about much is hormones. I mean, we'll talk about insulin to some degree, simply in relationship to our ability to digest carbohydrates, our ability to move those sugars into the cells, but we're not going to talk about them to a large extent. Storage proteins. Myoglobin is one example, storing oxygen in muscles. You have transport proteins. You have structural proteins, such as collagen. You have protective proteins, such as immunoglobulin. And you have contractile proteins that do the mechanical work of the muscles. Folks, all of those started with amino acids. And those amino acids have to be in a specific order in order for them to perform correctly. The amino acids have to be in a specific order in order for them to perform the task at hand. You change any order, you change out one amino acid, and you're going to completely alter what it can do. An example, oxytocin, which is used to smooth muscles during labor, to ease contractions during labor. Oxytocin differs from vasopressin, which opens up the blood vessels, by exactly 2 amino acids. Everything else is the same, but you change those 2 amino acids and you completely change its behavior. Okay, so we've talked about the fact that amino acids are made through these peptide bonds. You can get quite a few different peptides with the same amino acids, and these peptides do change how they, what they do based off of the order of those amino acids. With respect to proteins, there are 4 levels of structure: primary, secondary, tertiary, and quaternary. I have trouble with words today. Words are hard some days. We're going to talk about each of these with respect to our proteins. Okay, primary. We've actually already been talking about the primary structure. The primary structure is the formation of those peptide bonds. The primary structure is the order in which those amino acids combine. Again, as I said, the amine will always be written on the left-hand side in a primary structure. That is your N-terminal. And the carboxylate group will always be written on the right-hand side. That is your C-terminal. When we build up a primary structure, we're going to build up by adding to the carboxylate end. So if I were to bring in, say, glycine next, if I were to bring in the glycine next, it's going to add to the carboxylate end. So that when you combine these, what winds up forming is another peptide bond. And now glycine is the C-terminal end. And we would add glycine to our list of amino acids.
So it would keep this going like that, and then like you just put a dash after every amino acid.
Correct. The name is always going, now they may use the 1-letter name. I tend to use the 3-letter names, but you can use the 1-letter name, but you're going to continue adding to the right-hand side. And to say what it is, you literally say what are the parts of the protein. So again, your primary structure, we've actually already been doing is the formation of that peptide bond. You're always going to add to the carboxylate end to make the, to add a peptide to it, or an amino acid to it. Now, once you got this is the valine residue. This is the cysteine residue. This is the glycine residue. It's no longer just glycine. It's no longer just valine. And we label these residues in order. Questions about the primary structure? Trying to let people catch up with writing. So this is angiotensin 2, and it has to be, it's a 8 amino acid residue chain. So 1 out of 40,000 variations that you can have. 1 out of 40,000 variations of these 8 amino acids produces angiotensin 2. Only 1. If I change any one of these residues, you change the structure. You change what it can do. We will see just how important that order can be when we start looking at enzyme behavior. Because when we start you may have talked about enzymes in biology or something like that. You have the active site. If these amino acid residues are not in the correct order, whatever's coming into that active site's not going to bind. They have to be in the right order in order for it to bind in the active site. So for this particular one, you've got asparagine, arginine, valine, tyrosine, isoleucine, histine, proline, and phenylalanine. That has to be the order.
So would the variation come in like where you place the substituents or something?
So the variation would be take these. If I swap arginine and asparagine, that's going to be a new variation.
Oh, okay.
But if I swap asparagine and arginine, I'm not going to have angiotensin 2 anymore.
Okay.
I'll have something completely different. It's going to behave differently, everything. So on a test, one of the things that I tend to do, I know this from just, it's going to be, I will give you a 2, a 3, or a 4. Basically, I will give you the residue chain, tell you basically what is in it, and you have to draw it. You have to draw the protein. I can guarantee that's going to be a test question. I will give you the residue and you have to draw the protein. That's one reason I gave you the amino acids and gave you that sheet prior to spring break. And so you could start memorizing. Because you need to know the 3-letter code, you need to know the 1-letter code, and you need to know whether it's going at what pH it's going to be acidic, basic, or isoelectronic. So you know how to draw it. Okay. Now, we've talked about the residues and how you add residues onto the C-terminal end to make the primary structure. What we're going to get into next is we're going to start talking about interactions between this structure. And it's those interactions that make the secondary and the tertiary structures. Some of those interactions are hydrogen bonding along the backbone. Hydrogen bonding along the backbone is going to be responsible for the secondary structure. Some other interactions. And so where can you hydrogen bond? Well, you got lots of places to hydrogen bond. You got the amine group that can hydrogen bond. You got the carbonyl group that can accept a hydrogen bond. You got the carboxylate group that can accept hydrogen bonding. So there's lots and lots of places along the backbone where hydrogen bonding can readily occur, whether that's accepting a hydrogen bond or whether that is contributing to the hydrogen bond. You have lots and lots of places where hydrogen bonding along the backbone can occur. Another thing that will influence the shape is going to be hydrogen bonding of the R groups. So if you look at some of your amino acids, such as asparagine. Asparagine, if we go back, I'm going to go back real quick. I'll come back to this slide, don't worry. If you look at asparagine, asparagine has another carboxylate group on the side chain where it can hydrogen bond, as does arginine. Arginine has lots of places it can hydrogen bond on the side chain. Valine, however, does not, because it's got a nonpolar side chain. So when you're looking at these possible relationships, some of the R groups can hydrogen bond. Asparagine and arginine are two. Phenylalanine can, but it's going to be weak because of that phenyl group. You're also going to get ionic attractions, where you can form what's called a salt bridge. This is going to occur between an acidic and a basic side chain. So if at that particular pH you have an acidic and a basic side chain, you're going to form a salt bridge, potentially, between two of those side chains. When we start folding these proteins, you're going to wind up with hydrophobic pockets. That's where valine is going to come into play, or glycine is going to come into play. Those would form a hydrophobic pocket potentially. They're going to be a hydrophobic pocket because the side chain is nonpolar. So again, another thing you have to memorize. from that table that was 18.3 is which of those side chains is polar, which is non-polar, which is acidic, and which is basic. If you haven't started memorizing that table, I would get to it. You've got what you're going to be given on the test. Going through and memorizing the order and what it is going to be useful to you. Finally, when you have something like cysteine, you can form disulfide bonds, sulfur-sulfur bonds. Sulfur-sulfur bonds can make for a very strong interaction. And therefore it can make for a very stiff protein structure. So let's talk about the secondary structure, which involves hydrogen bonding along the backbone. Well, this is a picture of all of them. One of the opportunities that you have is you can take and make what is referred to as a beta sheet. Beta meaning bent is how it works out. This beta sheet comes from an interaction of hydrogen bonding with the backbone. In this diagram, the ribbon structure is the backbone. Ribbon is representing the backbone. You can have what is considered to be a beta pleated sheet. Which has hydrogen bonding between the backbone. That is an example of a secondary structure. You can also have an alpha helix structure, which this one represents a helical structure, an alpha helix. The difference is this is a side-to-side interaction of the hydrogen bonding between the backbone. So they have to be side by side. Whereas when we're talking about the alpha helix, notice that the interaction is every fourth amino acid. And so it's wrapped upon itself, and every fourth amino acid is hydrogen bonded to itself, to the next one, to the fourth one away. I like this diagram because this diagram is good at showing you all the possible interactions. But again, the beta pleated sheet and the alpha helix represent secondary structures. If you would like a better picture of the beta sheet or the alpha helix, you can look at page 570 and 571 of your textbook. Those represent secondary structures. The third type of secondary structure is a globular protein. Basically, it looks like a thing of spaghetti on a plate. It's going to have some beta sheets. It's going to have some alpha helix, but it also has other things, which gets into the tertiary. So these two represent secondary structure. Secondary structure focuses on hydrogen bonding of the backbone. Secondary structure focuses on hydrogen bonding of the backbone. If there's lots of hydrogen bonding, you're likely to get a beta sheet or an alpha helix. If there's not a lot of hydrogen bonding on the backbone, or if it's willy-nilly, then it's a globular protein. Or seems willy-nilly. There's nothing willy-nilly about proteins. When we're looking at the tertiary structure, what we're looking at is how do the side chains interact. So again, secondary structure is all about the backbone. Tertiary structure is how do the side chains interact. That's where you can have a water-free pocket. If you notice, all of these are nothing but carbon and hydrogen. When you have nothing but carbon and hydrogen, you got a nonpolar pocket. Nonpolar means you don't like water. So you form this nonpolar pocket where you can't have water. You will notice that this tends to be on the inside of a tertiary structure. The reason it's on the inside of a tertiary structure is because what is your body mostly made up of? Water. So these aren't going to be on the outside. They're going to be on the inside, forming these little pockets. By the way, this particular diagram is on page 572 of your textbook. I told you I needed to bring in pictures today. I think the pictures speak more than I can draw. So that's why the pictures come in. So you can form hydrophobic pockets where you're not going to have water. You can also form hydrophilic pockets. You'll also have your hydrophilic side chains on the outside of your protein. That way they can interact with the water. But you'll form hydrophilic pockets where you have polar side chains. You can form salt bridges. Salt bridges are going to occur between an acidic side chain and a basic side chain, or a positive and a negative side chain. I mean, after all, what is a salt from your previous course? It's something that had a positive and something that had a negative. We're talking the same thing here.
You're still talking about tertiary structures, right?
This is still tertiary structures. And you can get that disulfide bond I was mentioning. Disulfide bonds form a strong interaction. But you only have a couple of options to form disulfide bonds. There's only 2 amino acids that have the sulfur in place. And cysteine is the primary one, because cysteine has that sulfur right there on the end. So let's back up a moment and let's talk primary through tertiary. Your primary structure is the order in which your amino acids combine to form a protein. Your primary structure is the order in which the amino acids combine to form the protein. The primary structure is the order in which the amino acids combine to form a protein. Your secondary structure is the result of hydrogen bonding along the backbone. The secondary structure is about the backbone, in other words. It's about that portion of the protein. Secondary structure is about the backbone. Specifically, hydrogen bonding along the backbone. Does it make the beta pleated sheets or does it make an alpha helix? What does it do? It's about the hydrogen bonding along the backbone. That is your secondary structure. It's about the hydrogen bonding along the backbone. Your tertiary structure takes into account the bottom four interactions. It's about the side chains. What do the side chains do? Your tertiary structure is about the side chains and how they interact. Does that help specify what you're looking at? Because it all combines into your protein. And it all determines how your protein is going to behave. You change the order, you completely change the identity of the protein. You change how it's folding, you completely change how it can interact with other molecules. This just goes into more detail. Secondary protein describes the spatial arrangement of the polypeptide backbone. Again, it's all about the backbone. Is it alpha helix, a beta sheet, or is it a globular protein, which means that there is some fibrous sections where the backbone has interacted, but there's a lot where it hasn't. Oh, I did show you the alpha helix. It's just not in the order I had planned. This is the alpha helix. Again, it forms a helical structure. And this is the nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen. And you'll notice then that the hydrogen bonding, so if you got the nitrogen, carbon, carbon, every third one is a double bonded oxygen. That interacts with the nitrogen. Double bonded oxygen interacts with the hydrogen, with the nitrogen. So it forms that alpha helix. This is the beta sheet. Again, you have nitrogen, carbon, carbon, nitrogen. Carbon, carbon, nitrogen. The difference is this wraps back and forth and it forms a flat folded sheet. That's why it's called a beta sheet, a pleated sheet. These are examples of the secondary structure. And then globular proteins, like I said, are a lot. They do have some internal hydrogen bonding, but it's not as structured as the beta sheet or the alpha helix. They appear to be folded in an irregular manner, but it's not. The way they are folded is important to their behavior. And yes, I will start, if I'm going to use a lot of pictures, I'll start posting the PowerPoint in advance. Fibrous proteins. So these are the proteins that have the beta sheets or the alpha helix. They are very fibrous. Includes keratins, collagens, elastins, myosins, and fibrin. Whereas your globular proteins, which appear to have less structure, include insulin, ribonuclease, immunoglobulins, hemoglobin, and albumins. Those are just examples from the secondary protein structure. Tertiary structure, as I said, depends upon the interactions of the side chains. They maximize stability based on their side chains. They are governed by the interactions of their side chains. You can have a mixture of everything. Disulfide links, connecting loops, beta sheets, alpha helix, when you start talking about that tertiary structure, because it's all about their side chains. This is more pictures of tertiary structures. It's all, in this particular case, it's all about how they have folded together. This is just one this is one protein chain, and then it interacts. And like I said, this looks to be an enzyme. If you did not have the correct proteins along that active site, what happens on the inside of that active site would not be able to take place. Quaternary structure or quaternary. Basically, folks, a quaternary structure, which is the only one I haven't talked about so far, is how more than one protein interacts. So primary, secondary, and tertiary structures is about a single protein. Primary, secondary, and tertiary is all about a single protein. Quaternary is about more than one protein. Primary, secondary, and tertiary is about a single protein, whereas quaternary is about more than one protein. So what is primary about? What are we describing when we say the primary structure?
The order of the amino acids.
The order of the amino acids. What are we describing when we describe secondary structure? The backbone. How is it? Does it hydrogen bond to form an alpha helix? Does it hydrogen bond to form a beta sheet? Or is there some form to it, but it's not obvious, which means it's a globular? What are we talking about when we talk about the tertiary structure? The side chains, how they interact. Do they form a non-polar pocket? Do they form a polar pocket? Do they form a salt bridge? Do they form a disulfide bond? Disulfide bonds help strengthen globular proteins in particular. And then what are we talking about with a quaternary structure? More than one protein interacting. An example is heme. You have more than one protein that interacts. The purple represents one, the yellow represents one, the blue represents one, and the green represents one. With the iron in between. They all combine to form a quaternary structure. Okay, so we've talked about making the protein. That's all we've been talking about is how to make a protein from the amino acids. The last little bit of chapter 18 talks about how to destroy a protein. Folks, the very first thing is the same thing you learned in chapter 18 or 17. You can hydrolyze. In other words, you can add water. And if you add water to a protein, you're going to destroy the peptide bond. You're going to take the peptide bond and you are going to break it. So if I want to break the peptide bond, I can add water. Now, it's not as easy as it was for the organic molecules. Otherwise, we'd be in dire straits biologically. It has to require a catalyst to make it happen. And that catalyst is digestive enzymes in your stomach. But we can hydrolyze by adding an acidified water. We can hydrolyze our protein back into its amino acids. That's one way we can destroy a protein and its function. Is we can perform hydrolysis. It's going to be in an acidic solution. It's going to involve water in an acidic solution, and it's going to require a catalyst. Otherwise, we would be in dire straits because we wouldn't have any protein. So this is a little bit different than what we were talking about with the organic molecules for hydrolysis of an amide. In that, you have to have a catalyst. Hydrolysis of an amide back in chapter 17 just required H3O+. In this chapter, you've got to add an enzyme to make it happen. Because I want to give folks time to write this slide down. We'll pick up with this information on Thursday with the last thing, which is denaturation. And then we're going to start moving into, I want to say we actually get into we'll talk briefly about enzymes. And then we may get into the, well, I say briefly, we'll probably spend most of Thursday on enzymes. Any questions for me?
I'm just curious, what chapters will the next steps be?
18, 19. What did we go over in 19? Probably 18, 19, and 20 is what I'm guessing. It just depends on, I gotta look at when the next step is for one thing, and I gotta clean up one of my goals today is to clean up your blackboard a little bit, and I'll add today's PowerPoint into it, and I'll also add the next PowerPoint into it. Because when we get into biological molecules, I think the PowerPoint makes it easier to see. And that way, if you want to print off the PowerPoint and take notes on the PowerPoint, you can. Okay, I will see you on Thursday. I hope to get another one of your labs graded sometime, or one of your labs graded between now and Thursday. I'm trying to keep up with this class, but it's hard some days. Normally I use my conference is not during spring break, and I use my spring break to catch up. Wasn't lucky that time.
So the next class period, we'll go over the beginning of chapter 19.
Yeah, we haven't got, I don't forget your own phone.
That's not.
Oh, that's not yours.
No, that's
I
not
will
there.
take it to the office. Yeah, we'll finish 18 fairly quick because we're really just talking about the new trades and the studies, and then we'll get into and what can happen to do nature proteins, and then we'll get into.
So I missed lab yesterday, okay? And it was for a family emergency last Thursday. I had to go out of town for family. My uncle was not doing too well. So I was wanting to know if I do the food pantry extra
Right.
credit, would that cover the lab?
I mean, it'll cover the points you missed from the lab, yeah.
Okay, thank you.